name is Taneshia Harmon. I'm going to be talking to you about light-dependent reactions in the Calvin cycle. But before we start, we have to both start off. I know this is kind of like a bad picture, but this is my drawing of the chlorophyll, which is in the leaves. And most photosynthesis will take place in the leaves. CO2 needs to come into the leaf through the guard cells, which allow gas exchange. The chloroplasts are actually in the mesophyll of the cells, which contain the stroma, which is this space right here, the thylakoid membrane, which is this individual space there, this thylakoid space, which is going to be the space in between the thylakoid membrane. You have the inner membrane, the inner membrane, and also the outer membrane, which is on the side. Each cell will have about 20 to 50 of these chloroplasts in them, and they allow for the photosynthesis, reflecting the light and absorbing light of that sort. So we know the photosynthesis equation to be CO2 plus water and light energy produces glucose and oxygen. So, like I said, we're going to be talking about the light-dependent reactions, which is also known as the light reactions, and the Calvin cycle, which is also known as the dark reaction or the CO2 fixation. So, I did my little drawing here of, the reaction center, just to give you an idea, of this photosystem. And again, the photosystem contains the reaction center and the antennas, which is just like a collection of pigments. So you'll have the chlorophyll in here. You have the carotenoids, I think I'm saying that right, which is the red pigment, and the chlorophyll being the green pigment. In the center here is the reaction center, which contains the reaction center chlorophyll A. And the reason because on the outside here it's also chlorophyll A, but this is like special type of chlorophyll A, and the reason that they're so special and what sets them apart is that their position. They absorb the energy by the resonance energy transfer. So you'll have the energy coming from right here. It'll be bouncing from these chlorophylls to this chlorophyll to that chlorophyll, which in turn transfers down here over into the reaction center. And that way, that's where the energy is transferred. So from understanding this somewhat depiction of this photosystem, this is what takes me to the light reaction. So, again, please bear with my drawing here. We'll be starting over here with photosystem 2, and as you can see, the light comes in to the chlorophyll here, which they send their energy into the reaction center here. The chlorophyll A, which donates its electron to the transport chain, which is going to come through here, and this is where the proton is going to move through the gradient across into the thylakoid space up here, which results in the higher concentration of the protons. I also like to add back, I know I'm skipping a couple steps, but over here in the ATP synthase, this is where we're going to make the ATP. So that'd probably be like the last step. But back over to the photosystem 1, the previous millimicrosecond in the reaction center chlorophyll A donates its electrons to ferredoxin, which donates its electron to the NAD right here, which makes the NADP pH. In photosystem 2, you have the water splitting up top, and oxygen is given off as a product. And with this water splitting, the electrons are replenished because, this down here is going to end up going off into the Calvin cycle is ADP. So, I mean, there has to be a way for, the electrons to be replenished for the cycle to continue on and on. So, yeah. The ATP here is going to be was going to be sent to the Calvin, so it's really not doing any work in this process. So that's why the water splitting is so important in the photosystem too, so that the electrons get replenished, so that the cycle can continue on and on and on. Which brings us to actually the Calvin cycle. The Calvin cycle takes place in the stroma. The ribisco is the enzyme used to fix carbon. The plant takes CO2, adds it to a 2,5-carbon molecule, the ribulose 1,5-bis phosphate. I think I said that correctly, in the stroma. So the ribisco comes in to catalyze the reaction, which makes this unstable 6-carbon molecule, and it's like pretty much as soon as it makes this 6-carbon molecule, it splits to make 2 molecules that are 3-carbon, and they're actually really stable as well as identical, and we know these as the 3-phosphoglycerate molecules. So this is what's taking place right here. You have the CO2 coming in, and then it's on this side. Then you have the 5-carbon ribulose 1,5-bis phosphate. The plant throughout the cycle, the plant uses ATP and NADPH here, and the NADPH comes from the light reaction. So then after all that takes place, it comes down here, and the glyceraldehyde 3-phosphate exits out, which is down here, to make sugars, fatty acids, amino acids. And we started off with 2 3-phosphoglycerates, and now we only have 1. So really on this side of the cycle is when all the fancy chemistry gets done. Pretty much the cell is going to rearrange things and, end up making the ribulose 3-phosphate, and it takes the ATP now, makes ADP, and now we start the cycle all over again. But really, to understand the Calvin cycle, we have to remember that it turns 3 times. So 3 molecules of the CO2 combined with the ribulo BP, I think that's what I said, end up making 3 molecules of the 3-phosphoglycerate. So in turn, we're like, once we get back over here, we've made like 15 carbons, but we started off making like 18. So that's pretty much Calvin cycle summed up. Seeing that the Calvin cycle is so complex, it's kind of difficult for me to explain, especially when we get over here to this side with all the chemistry and stuff like that. It's very complex. But that is light reactions and Calvin cycle in Jiffy. Thanks.